Dr. Abdelhak, thank you for joining us on MD Newsline podcast. Can you please introduce yourself to our audience?
Absolutely. Thanks for hosting me. I'm Ahmed Abdelhak. I'm an assistant professor of neurology at the University of California, San Francisco. I'm an MS clinical scientist. I've been working in the MS field for 10 years, and I'm a huge biomarker fan.
Yeah, you did a great presentation today on fluid biomarkers, and I would really like to explore that a little bit. So tell me, what are some of the most important recent advancements in fluid biomarkers for MS progression, and which markers do you consider most clinically promising for risk stratification?
So we are having a really exciting time now. There are many advancements in the field, as you mentioned, and we are, as a whole field, moving to try to understand progression better and treat it better. And of course, what you can't monitor or measure, you can't really treat. So we are very excited about biomarkers of progression.
Mm.
Um, some of the most exciting data we are having are for markers that we know, like neurofilament light chain, but also the emerging data about something like glial fibrillary acidic protein, GFAP, or chitinase 1, or other forms of chitinases are very exciting. And we have also emerging interesting data about synaptic injury and how it can contribute to MS progression. Taking all of those together, you can really start seeing we are trying to look at the brain as a whole. We are trying to understand what's happening in different components together, even if we are using single markers or moving to biomarker panels.
Interesting. So you mentioned some biomarkers already. How do glial markers like GFAP and CHIT1 differ mechanistically from established markers like NFL, and what do they reveal about MS pathogenesis?
Wonderful question. And I think the first step to answering that will be to look which cells are releasing those biomarkers or what's the origin of those biomarkers. For something like NFL, we know very well it's coming from the neurons, from the axonal structures. We know if you put neurons under stress, they will release NFL. So ultimately, anything in the MS pathology that will stress the neuron or damage them will result in higher NFL values. If you look at glial fibrillary acidic protein, it's a slightly more complex situation because we know that GFAP is a protein that's present in the astrocytes. And we actually know that astrocyte damage or injury in MS is not very pronounced. Can happen, but not very severe. So the question is how an intracellular marker like GFAP is released from the astrocyte and what would it mean? Our current understanding suggests that GFAP is released based on astrocyte activation. If you have large number of active astrocytes, you will see higher values of GFAP in the blood. Chitinase 1 or chitinase 3, like 1, those are two different markers. It's also very promising markers. They are coming from another component in the brain, which is the microglial cells. So microglial cells or myeloid cells in the brain, you find a lot of chitinases there, and they are known to release those chitinases. 1 or 3, we are still, not sure that we know exactly how differently they are involved in the MS, but we know that for both markers, if you are measuring them in the blood, we have to be really careful because some of the blood cells are also releasing those chitinases. So you will have some noise in the blood. That doesn't mean that those markers are not informative. It's just important to understand the real source of what we are measuring. So mechanistically, based on that, every marker is reflecting to you what the cells that are releasing those biomarkers are doing. So when we are measuring NFL, we are talking about neuroaxonal injury. GFAP, we are talking about astrocyte involvement. And for the chitinases, it's reflecting what the microglia are doing. So you can measure all of those together and get a more holistic view on what's happening in the brain in a patient with MS.
Interesting. How do serum GFAP concentrations relate to MS lesion pathology and progression? And is there evidence for GFAP as a marker of glial activation and severity?
That's a complex question. So how GFAP would relate to MS lesions? We know that if you have the so-called gadolinium-enhancing lesions, you have high GFAP values. For a simple reason, GAD-enhancing lesions mean disruption of the blood-brain barrier, and an integral part of the blood-brain barrier is the so-called astrocyte infeed. So when you have a GAD-enhancing lesion, you have pathology in an area that directly relates to the astrocytes. So we see high GFAP value in this situation. However, it has to be clear that the GFAP value we are seeing with the GAD-enhancing lesion don't go that high as NFL values would go. So they are showing you still two different signals.
Mm.
About the progression, what we have been seeing so far is MS patients who are having high GFAP value, they are tending to have more progression, higher number of the so-called PIRA events. And there were some very interesting data showed yesterday that if you even use machine learning approaches to cluster your MS patients, you will see the group that have high GFAP and rising GFAP concentrations is the group that is going to suffer from most of the progression over the follow-up period.
Mm. What's the significance of combining GFAP and NFL levels and predicting progression independent of relapse activity? And how should clinicians interpret high versus low levels of these proteins?
That's another amazing question. So combining them is a little bit tricky. So you have to know first they are showing you different signals. That's number one. And number two, that from what we know so far, high NFL and high GFAP is really showing you high progression in patients who have earlier stages of the disease, like relapsing remitting MS. If you go to patients who are late in their course, like with secondary progressive MS, we actually saw GFAP to be more informative or more predictive for progression in subjects who have low NFL values. It's a little bit of a paradox that we are still trying to decipher to understand more what is unique about this population that makes this constellation of high GFAP, low NFL to be particularly predictive for progression. So measuring them is very important. Together is very helpful. If you have disease activity, the values you are seeing are most likely coming from the disease activity. But if you are measuring them in patient with very low likelihood of disease activity, like with our B-cell depleting treatment, then the high values are likely reflecting high likelihood for developing disease progression.
So, to what extent can things like fluid biomarkers such as kappa, free light chains, serve as a substitute for CSF bands, and what are the technical or regulatory barriers you see in their routine use?
So actually, the kappa-free light chain is now part of the MS diagnostic criteria. So we shouldn't be having a lot of regulatory issues using them. There are actually a lot of very well validated assays. I think there are a lot of data trying now to really pinpoint what would be the optimal cutoff value, after which you can say, oh, those kappa light chains are really helpful in diagnosing MS. We have a lot of very promising data. It's recommended that we use them now as part of the criteria, but certain labs will have different cutoffs. And I think some of the efforts out there is to unify the cutoffs and try to validate that in other studies. So kappa-free light chain can be very informative to a certain degree, very similar to how oligoclonal bands are informative.
Interesting. Can you talk a little bit about the results and statistical power of multinational analysis of serum GFAP and NFL and related biomarkers in predicting confirmed disability worsening and long-term outcomes in progressive MS?
Yeah, I think this is the key now. So those biomarkers, as helpful as they are, um, their meaning will be different based on your patient population. So if you are having a small study in your center, you might see a signal that's different than what other people saw, or you might even not see a signal. This is why it's really crucial that the different centers combine efforts. And we have actually some examples that were very successful in that, measuring NFL and GFAP in very large patient populations, showing how with this, how the combining the efforts really improved our predictive value using those biomarkers. After you combine those efforts and you measure those values in thousands of patients, it's another challenge that we are working on now, how you are bringing the meaning of that to individual patients. But this is a very active area of research.
Yeah, that's a very exciting area for me, especially when we look at these large data sets and the use of machine learning. How is machine learning or AI impacting that type of study? And what are some of the challenges? Because, obviously doing things across such a large data set, you come across certain barriers. So what do you think about machine learning and then
Yeah,
some of the challenges of making sure that things match up?
it's a very exciting field. There are so many approaches that you can use those AI-based tools and machine learning to accelerate our research. I think what I find very exciting is how you can use now some AI tools to harmonize the data or extract the data from more noisy cohorts. This is very helpful that like, you would otherwise spend hours and hours trying to do that, and maybe even longer. And having an AI tool that can do that quickly for you is a very helpful tool. When we then move to the machine learning approaches, we need to have then really large data sets. And we will then see maybe some clusters, as we saw in some of the presentations now. The question would be, what's the biological meaning of those clusters? So I think one crucial step here, we need to see after we identify those clusters, do they really make biological sense? Are we maybe just seeing signal artifact? Are we seeing an artifact related to certain noise or bias we have in our cohort? So we would definitely still need to go back to what we know about the biology of the disease and try to understand those clusters in the context of this biology.
Do you have any thoughts on maybe the future of making connections across specialties? Because we go to these conferences a lot, and I know that doctors are busy. They're going to conferences, they have papers, they're doing research, and everything like that. But since the body is kind of a system, and we have these ideas around, maybe things crossing the blood-brain barrier. This is the idea that inflammation or immune response might start in one part of the body, and maybe influence or make the body more heightened to have an inflammation or whatever response in another part. So do you have any thoughts on working across specialties at all, maybe across specialty studies and things like that?
So this is a very important direction now in the MS field, that we move beyond just looking at the brain and the spinal cord and really see our patients with what matters to them most. And if you think about the MS biology, we know that infections beyond the central nervous system play very important role in the pathogenesis of MS. We know actually data about the microbiome of the darm in our intestine, and how it can influence MS risk, and some emerging data about activity and progression. So we definitely need to take a look at the whole picture. We are already having a lot of cross specialty collaborations. I mean, the role of MRI in MS and how valuable is collaboration with neuroradiologists. Also, a lot of MS patients might have other autoimmune conditions, other systemic autoimmune conditions that would require collaboration with other specialties. So there are definitely some efforts there, but it has to be expanded more. I think we need to involve, for example, more of the physical therapy approaches. We need to make that integral part of our treatment of our MS patients, finding ways to address the challenges that our patients are seeing in their everyday. Those are all very important components.
That's great. Are there specific biomarker cutoffs or scoring systems that clinicians should be monitoring? And how are age, sex, and comorbidities accounted for when interpreting biomarker data?
So going back to those fluid biomarkers, like having those variables is very important to understand. So we know if I take healthy people and measure their NFL values, I will see that their NFL values is increasing with age. It does not mean that they have any neurodegenerative condition or MS or anything else. This is just how NFL changes with time. So a normal value for 50 years old patient is very high for a young patient. So in this case, you would need to always adjust for age for NFL, for example. For GFAP, we know as well that biological females will have higher values compared to biological males. So we will need to definitely consider sex as well. Body mass index is another important factor. After you use all those together, the best approach we have been having so far in combining those variables is using the so-called Z-scores. With Z-score, you are literally reporting how the value in this patient will deviate from the value in a healthy individual that would have the same age and sex and body mass index. And defining cutoffs of the Z-score is something of interest we are working on now. I think we see good results for elevated Z-scores, like a Z-score of 1 or 1.5, for example, for prediction of disease activity using NFL. But we might end up needing to develop specific Z-score cutoffs for every biomarker and potentially for every treatment group.
Mm. How do multimodal biomarker panels, combining fluid imaging and genetic data, for example, improve prediction compared to just using single fluid biomarkers alone?
Um, we are starting to explore that. So I think this is the area where the AI and the machine learning would come into play, because it's very challenging to use that with classical statistical approaches, leveraging all those multi-omics data, also with the very extensive clinical data. We have MRI data. So it's a very exciting area in the field. A lot of people are working on that. We are facing some methodological challenges, but I think the initial results are very, very promising.
Interesting. And just thinking about the fluid biomarkers, that you know right now, the or analytical approaches, which one do you think? What do you think can most dramatically change MS progression monitoring over the next 5 years? If you were to make a prediction, which do you have your eye on?
Uh, Very interesting. I think we are very close to having an understanding on whether GFAP will be helpful on individual patient level for predicting progression. So I think that's something that we should know in the next few years. But I think what would be more interesting in the next few years will actually be discovering new markers. So I think in the next 5 years, we'll be having way more candidates or even panels of candidates for biomarker that can predict progression. We'll have a longer journey after that, trying to really bring them to the patients. But I think we are starting this discovery era.
Are there biomarkers or biological pathways, especially that are under recognized, that you believe deserve more research focus, either in identifying early progression risk or identifying new therapeutic targets? Are there any that you are like, if I were to really shift resources in that direction, it would be?
So a good example for those are the microglia. So we are understanding from different aspects, pathology, histopathology, MRI, and body fluid biomarker, that microglia are playing a really important part of the disease. And you see how now we are having treatments like the BTK inhibitors that might be coming soon to the market, influencing those microglia and changing the disease course. I think the astrocytes might be understudied, in my opinion. We do see very interesting results for GFAP. There are some interesting data from different animal models showing that astrocytes in themselves can be toxic to neurons, causing neuronal injury. And so far, we are really lacking any reliable treatments that would directly impact astrocyte function. Some of the existing MS treatments will have indirectly an impact on the astrocyte, but it's not so far one of our main therapeutic targets. So this is definitely an area that, in my opinion, is worth further work in the field.
Mhm. Do you see potential gene editing being relevant in this field in any way in the future? Because we've seen gene editing come into play
Yeah.
with a lot of things, and it seems to be getting more. So, what do you think about that?
Well, while my research is not necessarily heavily involved in genetic, I
Mm.
think we can agree that there is no gene that is causing MS. We know that there are some genetic changes that are associated with higher risk of developing MS, but they do not necessarily initiate the development of MS. There have been very exciting work over the last 2 years, finding some SNPs that are also associated with a more severe disease course. But I think I will be very surprised if we end up using gene editing to manage MS. I don't think we have the target for
Mm.
that in comparison to some other neurodegenerative
Mm.
conditions that are really influenced by gene changes.
Yeah, be interesting. I always wonder because you have this. I was talking to someone about Epstein Barr earlier, and so this idea is that like everyone, a lot of people, most people we can say have it, but everyone who has MS usually
Yeah,
has pretty much has it 100%. So there's clearly some sort of underlying process that maybe might make people more susceptible. We don't know. And so it'll be interesting to see what we discover moving forward, why some people react that
yeah.
way or what's going on in their processes, or maybe it's not,
It's definitely a combination of things.
Mm.
You need to have the genetic predisposition, you need to have the EBV infection, you need to have other environmental factor, for example, low vitamin D level, for the disease process to start. It's I will be very surprised if by removing the genetic component of that we will not see MS anymore. I think this is very unlikely.
Mm, interesting. So when you look at the progression of personalized medicine, for example, what do you see progressing more in the field of MS? How do you think that we're going to get even further personalized than we are now?
Wonderful question. We are in a stage where we are really having a lot of treatments now, and we are using, for example, those machine learning approaches to identify clusters within our patients beyond what we already know. So we know that there is relapsing, there is progressive MS, and even with the new criteria, we are kind of more harmonizing our definitions. But there are potentially some hidden clusters within our patient population that could explain why some patients would respond to a certain treatment better than the other. So I think this is an area that can be largely accelerated by body fluid biomarker research. And we have presented in last ICTRIM some very interesting data showing that individual biomarker trajectories after starting a certain disease modifying treatment would actually tell you something about how good the patient is responding to this treatment and whether this patient is going to have high or low risk of PIRA in the future. So it's an area of a lot of progress, and we are having finally some tools that can inform us with that, like large biomarker discovery panels, the AI-based approaches, machine learning that can combine all those large data sets.
What was your biggest takeaway, or what were you most excited about your fluid biomarker presentation that you gave?
Uh, Well, I think the exciting part was after we gave that presentation showing what biomarker we think are very relevant for progression. We had 2 or 3 talks after that showing the real application of those biomarkers and how they really reflected progression in different cohorts using different approaches. What I found also interesting that we started to see some biomarker panels that were based on discovery tools and how they can help us predict PIRA. We are still very early in this field, but I think we will see a lot more of those examples in the next years.
Mm. In your experience, what misconceptions do clinicians hold about interpreting and applying fluid biomarker information in progressive MS, and how would you clarify these points?
I think one very important limitation that I see when we start using those biomarker in progressive MS, based on the data we have, even if we don't necessarily have an explanation for it, the meaningfulness of one biomarker is different in primary than secondary progressive MS. I think this is a key importance if you try to use NFL in secondary progressive MS, especially non-active secondary progressive MS, to predict progression. You are probably not going to see anything. While if you try to use NFL in primary progressive MS, NFL is highly predictive of progression in primary progressive MS. It's exactly the opposite when you use GFAP. So I think it's a limitation we have to keep in mind at what stage is our patient and is the biomarker we want to use the right biomarker for this stage.
Mhm. Interesting. What would you leave your fellow physicians with as far as words of encouragement? What you want them to focus on when they interact with their patients and things like that? What do you want to see more of in the field or in the research?
I think it's very important in the field to expand on the point you mentioned, is treating the patient as a whole, really making sure we are helping the patients with the challenges they are seeing in every day. And we have some examples for amazing efforts on that at UCSF and in other institutes as well. Um, from a biomarker point of view, I think we need to communicate with the patients what those biomarkers mean. There are certain different assays available now, and the patients might have access to those different assays. Some of them are approved in the US, some of them are approved in Europe. We need to tell the patient that a high biomarker value is not very straightforward to interpret. It needs really some expertise in understanding that the value from one platform is different than the other. So we are trying to use those tools, but I have to say we are still also trying to understand how to bring this knowledge to the individual patient.
So it seems like a matter of communication, like having a standard way to help patients understand these factors so that they can understand their own disease themselves? Or
Absolutely.
yeah.
Like if the patient would measure NFL, or if the patient would see their NFL values during an acute relapse to be elevated, we have to explain to them that this does not necessarily tell us a lot about their progression risk because it was measured during relapse. But what it might tell us is that they had an inflammatory event, and we need to make sure that the inflammation is suppressed in their cases, and that those biomarker values, they take time to change. Like if you measure NFL now, they are elevated, and you start a new treatment, you wouldn't see the NFL values to be low in a week or two. It will take probably a couple of weeks until they are notably lower. So we need to communicate those things with our patient when we start applying our biomarkers in everyday care.
Do you have any words for patients as far as what you would suggest for them when it comes to advocating for themselves if they've been diagnosed with MS? What are some of the places that you think that patients could use more advocacy?
Um, I think it's very important for the patients to know that they should really discuss everything that matters to them. There are some hidden MS symptoms that maybe in the busy clinical setting are not addressed substantially, like cognitive symptoms, like depression, like fatigue. And the fact that the patient is stable, for example, regarding disease relapses, they are not getting any relapses, does not mean that the other symptoms are not important. So it's really important to bring that to your physician if they didn't discuss that. And I know there is very low, like growing awareness in the field of MS physicians now that we need to actively address those symptoms, but patients should advocate for that as well. And there are very good examples from the National Multiple Sclerosis Societies with specific funding mechanisms for symptoms that are really relevant for our MS patients and for patients' well-being.
Wow. Is there anything else that we haven't covered? I feel like we've
I think we covered amazing set of
range.
things, including body fluid biomarkers and MS care in general.
Yes, thank you so much for joining us on MD Newsline. It's been a pleasure.
Wonderful.